and then I talk about strong, so first weak additive noise, then strong additive noise. And then we come to general anesthesia. That is an application. There are other applications out there, but this is one I find very, very interesting. So additive noise, so is supposed, yeah, well, let's put it differently. So multiplicative noise is known to change the stability of a nonlinear system. So this is well established. There are many textbooks on that. And additive noise first does not change the stability of nonlinear systems. This is not known. So this is Lyapunov stability theory. And there's a theorem on that shows that in linear systems, you do not change stability, and nonlinear systems, under certain conditions, you don't change it. But I was just curious how to describe the bifurcation stability of a system or bifurcation structure when you have additive noise. And I was really searching in the beginning, I was searching a lot in the literature and how to do this. And there are different approaches, but no approach really, I found they were all special for special problems. And but I was looking for a general technique, something that I can apply to neuroscience. That was the idea. And at the end, I found one. And in fact, my former supervisor started developing this. And that was Hermann Haken in Stuttgart. And then I realized what is done there. And it was really fascinating for me. And I'll show you here just the major point. So let's assume that you have two nonlinearly coupled modes, so two equations. And one mode is subject to additive noise. So here you see this, so they look very cryptical. But at the end, what you have to understand is here, these are two nonlinear equations. They are coupled, nonlinearly coupled. And just one equation, U0, is subject to noise. And the question is, what is the probability density function of UC, so of the other mode? And this mode is unstable, becomes unstable, and U0 remains stable. So the question is, how does this additive noise in the stable mode affect at the end the dynamics in the unstable mode in UC? And one can show by center manifold techniques that at the end you can describe Fokker-Planck equation, so an equation for the probability density function for the order parameter UC. And when you take a closer look, then you see that there's a shift of bifurcation. So there's a term alpha TH. So this is an additional term that you have because of the noise. And this is proportional to the noise intensity. So this means that at the end, you shift your bifurcation point by additive noise. And this can be shown by center manifold theory, but only for weak noise. So really close to the bifurcation point. And then you can show it analytically that this effect is there. And so at the end, this is what you find. So in A, you find a normal, in this case, pitchfork bifurcation without noise. And then you add this noise, and then you see a shift of the bifurcation point. So you just shift it here to the right. And so this is a noise-induced shift. And that was amazing. And I was happy to publish this with André Longta in Ottawa. And it was really fascinating to see that this additive noise can do this. And that was in some way a starting point. And what I working on more on other problems, I realized that at the end, what is the mechanism? So the mechanism is that that additive noise in a multidimensional system becomes multiplicative noise by the backdoor. So you change, and this is really true. So you get multiplicative noise effects by additive noise, but only if the additive noise couples in through a stable mode. So, and that was interesting in high dimensional systems. So that was in this case in spatially extended systems. And so I had the crazy idea, because I was curious how this works. Let's do it in delay equations. So I was really curious to work on that. And so delay equations or delayed systems are high dimensional, are infinite dimensional. And I thought, okay, that should also work in delayed equations. And then Jeremy Lefebvre came into play, and we started working on this. And so at the end, what I show you here is this delay differential equation, stochastic delay differential equation. So it's nonlinear with a delay tau, and this is scalar. So, and there we have additive noise. And in fact, you can really show analytically by a senti-manifold theory that the Fokker-Planck equation reads like this, and you have additional terms by the noise, and this is the A1 shift and A3 shift in this case, and they depend on the external noise level. And then you can plot probability density functions, and then at the end you see these the colors here are for different noise levels, and there you see the characteristic shape of these probability density function change with the noise level. So you see it at this maximum, the maximum moves for different noise levels. And so this is also indication that that's really an effect. There's a strong effect by additive noise. And so, but this is can only be shown for small noise levels, and really, really small ones. And if you go to larger noise levels, you have a problem, because then you have to take into account more higher terms in the semi-manifold theory, and then this becomes really crazy. So it's not realistic to compute all these nonlinear terms. It's not practical at the end. And so, but at the end, one sees that additive noise in multidimensional systems becomes multiplicative noise. So that was a motivation for Jeremy and me to think about other systems where you change stability. And then, of course, we both worked in neuroscience, and we thought, okay, maybe it can also tune frequencies in systems. So if you have a network of nodes, and this system has a certain frequency, maybe additive noise changes the stability of this one, or even tunes the frequency. And so this brings us to strong additive noise. And here I show you the major results. So at the end, we assume a very simple dynamics, a network randomly coupled. So here you see, we have a pointer here now. Here you see the coupling matrix, this Wij in color, and it's randomly coupled, and we drive this by noise, external noise, and we were able to derive a mean field equation. This is very simple. It's given below, and then we studied the dynamics of this network. And here you see it here on the left, top left, you see the spiking network. So you can say each node has a certain spike. You can map spikes to each node, and there you see certain rhythmic pattern. And when you take a closer look at the mean, spatial mean of this network, you see that the frequency of this mean changes. So with noise. So if you have a low noise level, this is now on the right. If you have a low noise level, you have an oscillation of 10 Hertz, and you increase the noise, then it shifts to higher frequencies. And so this also, it's a similar effect as shown before. You change at the end the interactions between the nodes. You change the property of the system by additive noise. And at the end, this is understandable simply by this equation. So the mean field equation looks like this. You have this F, and this you can derive it analytically in this system. And this F depends on the D, and D is the noise level of the external input. And when you change D, then you change the transfer function F, and so you change the properties of your system. And so here's some simulations. So for different noise levels, so from left to right, let's see, what is this? So from left to right, you increase the noise level, and then you see you increase the frequency. And so in the lower row, you see that there one sees the transfer function, and for increasing noise level, the transfer function becomes more flat. So it's linearized in some way. But that's in some way that's a known effect in neural systems, in networks of neurons, that you see that the effective transfer function with increasing noise level just linearizes, and in this case changes the mean field frequency. So you have an increase of frequency by noise stimulation. Okay, but that's really interesting, because then we thought, okay, where can this happen, in the brain? So we have definitely this effect. Where's noise in the brain? And of course, you can say, the cortex and subcortical structures are driven by the brainstem and the reticular activating system. And one can assume, okay, this driving activity might be noise. And so when this brainstem activity increases or decreases, you change the noise level in the brain. And so the input to the cortex or subcortical structures. And so this might tune the interactions between the cortex and, let's say, the thalamus. This might trigger a change of frequency. And in general anesthesia, one sees this change of frequency. And so, so the idea was, we were curious whether we could explain some phenomena we know in general anesthesia by this changing noise, by this change in the noise level. So on general anesthesia, I would like to introduce this here. So before, in the very beginning, surgery was done by the hammer method, let's say. So patients were hit on the head. They got unconscious, and then some operation started, or you drank alcohol or something like this. But then 1846, there was a very first surgery under anesthesia. So this patient took an anesthetic for the first time, and then operation started. And this really triggered, that was in Boston that time, and this triggered worldwide a real wave of surgery. Even 1 or 2 years later in Europe and UK, they started doing surgery under anesthesia. And today, of course, it looks different. You have all these machines, electronic devices, and this is well controlled. But nevertheless, it's still a mystery what's really going on under general anesthesia. So when you go into surgery, of course, it's supposed to be safe, or it's said to be safe from the medical side, because typically what the drugs do to the patient under certain conditions, because it's medicine, so it's a classification science, let's say. So for this age, let's say a man or woman for this age, with this health condition, with this weight, with this body mass index, responds approximately like this. And so you give a certain amount of the drug, and then typically in 60 to 70%, it works fine. So, but there's still, as always in medicine, there's still a rest. And in this case, in general anesthesia, that's 30%. And an anesthesiologist told me that in about of 30% of the cases, it's not clear what's going on in the patient during surgery. On the, there's some behavior where the anesthesiologist doesn't know what's going on, but with much experience, typically, it's no problem, because then the anesthesiologist knows what to do in these situations. But they're honest, they say, well, we do not know what's going on, really, and how, and there are also after effects in patients. So some patients vomit afterwards, or have a decline of cognition, or have cognitive after effects. And so it's really severe. And even I can say this, my own mother was under eye surgery, and she woke up during eye surgery, and she realized that she was awake. She could not move, but it was really dramatic for her. And she told me this story 3 years after the surgery, and she was still aware of all the details during the surgery. And that was really devastating for her. And so these situations are really bad. And so these are the reasons why it's necessary to do research in anesthesia, and it's good to understand much better what's going on in the brain under anesthesia. So, but from a scientific point of view, when you really think about it, what's going on, you have a drug molecule that enters the blood, and by the blood-brain barrier enters the brain and affects different elements in the brain, like synapses or ion channels and so on. So you have a molecule that binds to other molecules at the gates, let's say the ion gates or the synaptic vesicles, change the properties, and this is the reason why you lose consciousness. But it's known what these molecules do to the single synapses or ion channels, but it's not known how the system, the brain as a system, responds to that. And so explaining the bridge between the microscopic level or at the synapses to the system level, and that's to behavior, EEG or behavior at the end. That's the task.
Axel, I might have a small question.
Yes.
So what are the conditions to choose one anesthetic versus another? Like you in the previous slide, you have propofol and isoflurane. So what guides the choice of those anesthetic in a given context?
That's just experience, because isoflurane and propofol, for instance, are different. So propofol is a very powerful anesthetic drug that starts very quickly. So you take it, and a few minutes later, let's say you lose consciousness, and you give it by an injection. That's one part. And it ends, so when you stop the infusion, it brings you back quite rapidly. But propofol makes a strong sedation, but you cannot do a full surgery under propofol because it does not block the pain very, very well. And it just, it makes you unconscious, but it's very good that you say so. I forgot to mention that there are 4 criteria to start a surgery. So you have to be unconscious. You should not feel any pain anymore. You should not move anymore. And there should not be any memory of the whole procedure. And so you need an anesthetic agent that provides all the 4 elements. And propofol mainly makes you lose consciousness, but works not that well it's not a good painkiller, and you can still move with propofol. And so you need typically you apply a mixture of all these. So isoflurane, for instance, is also very good, keeps you the unconsciousness very well, and you can move a bit less. You can move less than with propofol, but it's also not a good painkiller. Remifentanil, for instance, is a very good painkiller, but does not provide you a good sedation. So you do not lose consciousness that quickly with remifentanil. And so at the end, you get a mixture, a wild mixture of all different drugs to that you provide all these 4 features, and that the surgery can start. Okay, so at the end, so what are the observations? So what do these anesthetic drugs do? So they reduce the cardiovascular activity, they reduce the respiratory activity, and they reduce certain brain rhythms. So that's a physiological observations. And yeah, these 3 aspects are very important, but neuroscience just focuses on the brain rhythm, but of course we should take into account the cardiovascular activity as well and the respiratory activity as well. But this, in the moment, that is too complicated. So that is too complex. Also, there's a feedback effect between these. So the brain might affect, of course, the cardiovascular activity, but there's also feedback from the cardiovascular activity to the brain. But in the moment, this is too complex, and so people doing in neuroscience research and general anesthesia focus on the brain rhythms. So at the end, what do you have? So on one side, you have the microscopic action of anesthetics, where you have a change of synaptic properties, let's say, and on the other side, you have the EEG, so the macroscopic brain activity, where you see that this changes. And so I show you here some examples. So in the baseline, you have certain EEG activity, and when you increase, so from top to bottom, you increase the anesthetic concentration, and there you see there's a characteristic change of the EEG. So at the point of loss of consciousness, you get many more lower frequencies, high amplitude, lower frequencies, and then at the end, if the anesthetic concentration is very high, you get almost no EEG activity anymore, and then you go reverse. So you when you reduce the concentration of anesthetic drugs in the brain, then you go back and you gain your normal EEG activity again. So there's a strong effect on the EEG. And so one approach that I show you here is to model EEG based on the anesthetic concentration. But as I said, typically you have a wild mixture of anesthetic drugs that you give the patient. And so, but each anesthetic drug has a certain effect on different elements in the brain. So it's really complex. And in addition, there are interactions between different anesthetic drugs. And these interactions are not known. So you have it's a big mess at the end. And so the idea was, so first studies really focus on the effect of a single anesthetic drug on single types of synapses, and then try to explain the EEG. But at the end, it's well known that typically you have a mixture of drugs, and you do not know the on-site concentration of the anesthetic drug on different synaptic types. And so there's it's a hole in the knowledge, just in the knowledge of neurophysiological knowledge, what's going on. And so the idea was to come up with a, let's say, with a more general approach, or more abstract approach that might also explain what's going on. So first I would like to show you another experimental or data analysis study that is quite interesting in this context. So I met Michael Vibral at Frankfurt at the university, and he computes the transfer entropy and active information storage and differential entropy in certain brain areas. And I provided them with anesthetic data and asked, so in the prefrontal cortex, measured synchronously in the occipital cortex, and asked them, what can you say about the information flow between the two areas when I provide you some data? And they computed the transfer entropy and active information storage and differential entropy and found the following. So these are the results. They found that the transfer entropy, so these are two ferrets, two animals, and the interaction between the prefrontal cortex and the occipital cortex. And they found that with increasing anesthetic concentration, the transfer entropy decreased, and the active information storage increased, and the differential entropy also decreased. So at the end, what does this say? At the end, they found, based on the data, that increasing the information, the anesthetic concentration, the information transfer between visual cortex and prefrontal cortex was reduced. So there's less information transferred between the two areas. In addition, the stored information in visual cortex was enlarged. That means that while there's less flow of information between the two, there's more information stored in each area. And this explains why there's a reduced information transfer, because more information is stored. And in addition, this is consistent with this finding. The available information in visual cortex, for instance, is reduced. So at the end, you have the clear, based on these data, you have the clear picture that the brain under normal conditions transfers information and passes and loses information in each area, shares everything. And now you increase the anesthetic concentration. Now suddenly each area increases their own stored information and passes less information to other areas. So become more independent, let's say. So all areas do their own thing. They share less information with the surrounding, and this is why you lose consciousness. And this is in full line with a hypothesis of Tononi. He came up about 20 years ago, that the loss of consciousness is related to a certain reduced information flow between areas. And this is in full line with this. And so this is the finding in experimental data. And of course, we were keen to find this in a neural model. So, and here I show you an experiment of Purden et al. It's from in the Harvard group. So they worked a lot on general anesthesia and explanation of EEG features. And it's really amazing. They did an amazing job. And they studied the administration of propofol, the EEG in humans. And at the end, they studied the loss of consciousness. So what is loss of consciousness? What is consciousness? It's not something, let's say, philosophical, but there's a clear definition. Here in this context, loss of consciousness is defined as the disability to respond to external commands. So the subjects had a task to follow. In this case, the task was to respond to a verbal command, something like, what is your name? And then, or can you hear me? And yeah, press a button, yes, no, something like this, very simple. And then
I have a question for you.
Yes.
So there's a question in the chat. There's clarification requested. So what is the difference between stored and available information? I think these are words you might have used.
Yeah. Yeah. So in principle, the available information is the information that you could share or store. This is how what I understood. So the available information is the information, yeah, can be stored or can be transferred. And this fits in this picture, because even the available information that you could share or store is reduced. And in that reduced available information, the part that is available, then there's an enlarged part that is stored. And there's an even reduced part that is transferred. So, honestly, these are measures that have been developed for certain nonlinear systems. And this is the interpretation of these results. And the question is, can we, with this, based on these definitions of information, different information, can we explain the situation? And when we take these criteria seriously, we say, okay, it fits to the picture.
Okay, good.
Okay, so here in this experiment, it worked like this. The anesthetic concentration was slightly increased. So here you see the baseline individual subjects, and with the time, the concentration is increased in this subject. And then every time at each stage, it's counted how often does the patient respond to external commands. And in the beginning, where there's in the baseline, you see it's 100% response. So the blue and red lines are different command types. So it's clicks and verbal commands. And there you see it's okay. In the baseline, the subject responded quite well, almost 100%. But then increasing the anesthetic concentration, the number of clicks and verbal and responses to verbal commands decreased, and at a certain point, there were no responses anymore. This is defined as the point of loss of consciousness. And this is in this plot, yeah, it's plotted on the lower part, in the bottom part. So this is the definition of loss of consciousness in this context. So, and yeah, that's this. And when you measure the EEG, the frontal EEG in this case, and plot the time frequency distribution, then you see the following. So on the left, so horizontal axis is time or concentration axis. And there you see that at the very left, there's no anesthetic concentration, so baseline condition, and then you increase the anesthetic concentration. Then you see that at a certain time, there's a, let's say, beta rhythm at about 15 hertz comes up. And then this decreases slightly to about 10 hertz. In addition, and this starts at the point of loss of consciousness. So the beta and alpha rhythms start a bit earlier, before loss of consciousness, but there's also a 1 hertz, let's say a delta activity at between 1 and 3 or 4 hertz, and this starts at the point of loss of consciousness. So, and when you go back, so when you return to consciousness, so this is ROC, so return of consciousness, then you see a kind of reversal. Then, so you decrease the anesthetic concentration, and then this delta activity decreases and vanishes, and at the end, the strong alpha activity becomes a beta activity, and at the end vanishes. And that's a kind of smile. And so we thought, okay, that's a nice picture. And so that's a smile effect. And so in reality, it's not symmetric. So this smile is not, it's not symmetric between left and right, but nevertheless, what you see here clearly is that you have a strong alpha and delta activity at loss of consciousness, and this strong alpha emerges and starts a little bit before the point of loss of consciousness, but is very prominent. And our model focused on this alpha activity, and we thought, okay, can we explain this? But at the end, you remember our work on additive noise. We thought, okay, additive noise tunes the frequency in a system, but what happens in the brain when you increase the anesthetic concentration? You decrease the excitation level in the brain by reducing the activity of the reticular activating system or the brainstem. So there's a decreased activation of the brain by the reticular activating system. So reduced level of noise. And so we said, okay, let's just check whether we can explain this alpha, this induction of a beta and alpha rhythm just by changing internal noise level. Maybe
So we have a few questions in the
yes.
chat. So in the figure you have right now, so what does the solid white line means?
Yeah, that's the edge frequency. So that's a measure of it's a kind of boundary. It's just a visualization of boundary of the frequencies. So when you see it's just a transition, it marks a transition between activation and deactivation. So there's a definition of 90%, 95% edge frequency. You can look that up, and it's just for visualization that it marks the area where you have strong activity.
Okay, so basically like where the changes in frequency are statistically significant or something. Okay. There's a question, how this spectral change compares to sleep onset?
Yeah, sleep and anesthesia is quite similar. And in sleep, you also find this strong delta activity. But there are other features like this alpha activity. So you have spindles, you have alpha spindles, they also emerge, but at the end, they have different nature than the alpha activity you find in general anesthesia. But in sleep, you also find, you also find some alpha activity, increased alpha activity when you go asleep. You also find delta activity, but at a closer look, they have different nature. They have a different shape. They're in slightly different frequencies. And this brings up the question, so what is general anesthesia compared to sleep? Is it different or is it similar? Or so there's some similarities because the brainstem is also controls very strongly sleep rhythm. So there's a sleep switch, so it plays a major role, in fact, in sleep generation. But at the end, general anesthesia and sleep are different because you can wake up somebody who sleeps, but you cannot wake up a person who is anesthetized. So there's really there's another mechanism behind, a fundamental mechanism behind, but they are common features. Okay. So this is all, as far as I know, this is all that roughly that we know, that there are similarities or and differences.
Thanks, Axel. I'm the one who asked that question. So that means, however, that the noise story that you're going to present us here might actually carry over to some aspects of sleep. Perhaps.
Yes, but in sleep, so as far as I know, sleep is, there's a certain structure in the brainstem that is responsible for the sleep switch. Yep. And I think this is almost established that this area, it's a brainstem area, that this brainstem area really is responsible for the sleep switch. So there's more knowledge. I would say the research is more advanced in this context. I think it's established that this plays a major role, but in general anesthesia, it's not known whether this sleep switch really plays the major role. So there might be something else because, as I said, at the end, the response, the behavior during sleep and anesthesia is a bit different, or it's really different because you cannot wake up a person under anesthesia. but you can do this for sleepy person. And so I think the brainstem plays a major role, but this sleep switch might play a role in anesthesia, but it's not not the major element. But it's a good question. It's a good, yeah. Is there an effect? Can we explain sleep rhythms by this noise effect? So never thought about it. So I'm not working on sleep, so I do not know too much on the literature on that.
There's a clear gating effect. Yeah, there's a clear gating effect of information at the thalamus and sleep that gets shut off, right? So
yeah.
Information doesn't go on from the senses. Yeah.
So do you know of any noise effects, or is there?
No,
I don't know.
But you're right. I mean, there is a switch in the brainstem. Yeah. So
yeah.
it might be quite different. So okay, thanks.
Yeah. So there should be differences. So between the two, because the phenomenon is different.
I have maybe two small questions quickly before you keep going. So how this, I guess the figure you're showing here, how this compares to the figure on the first slide when you showed that EG becomes flat in anesthesia. So there was a figure early on where you showed that EG was becoming flat during anesthesia, and
EG becomes flat.
Yeah, you if you go yeah, right there. Sorry, you just skipped it. Yeah.
I see. Okay. Yeah. Well. Okay. So there are certain depths of anesthesia. So let's take this figure. So we have we have the baseline, and then there's 5 microgram per kilogram, and just about the loss of consciousness. You might say that's maybe the stage where you find the alpha. You find a strong alpha activity with a high amplitude. This is a situation where you have the alpha. But when you go to deeper anesthesia, then this smile shape vanishes, and then you get other features, because then you do not find this alpha activity anymore. This vanishes, and you only get the delta activity, and this is the flat line.
Okay.
And
great.
so in that figure here, this is this is still in a lab. It's not a real surgery. So these are the results of Purdon. And they ask students to do this under propofol anesthesia. That was not a deep anesthesia. It was a medium to light anesthesia. And so you still get this alpha, but not deeper. So if they would have gone deeper, then of course the alpha vanishes.
Okay, and then there's a question like, does this activity looks different in 30% of people who do not have full or typical anesthetic responses?
Yes. So so this very nice figure of Purdon, it's amazing. It's an average over subjects. And so you have to take a closer look at the paper to realize that this is not always the case. And when you talk to anesthesiologists, whether they see this smile effect, they say very rarely you see it in single subjects like this. So you see the tendency of this smile, you see it somewhere, but there are also patients where you do not see any strong alpha activity. But what you always see is the delta activity. So once I got just a random sample of patients under surgery, and in about half of the patients, I found this strong alpha activity here, this smile effect. But in others, they also lost consciousness. I did not see, obviously, that very strong alpha. And so it's not always like this. As typically in medicine, all patients are different, respond differently to a propofol level, and that's also a problem in comparing these different data, because typically all patients got another mixture of anesthetics. You see, they went under different surgeries. Some got heart surgery, others got an arm surgery, or what was light anesthesia, others were deeply anesthetized. And so, and the mixture is always different because in practice, the anesthesiologist adjusts the anesthetic concentration according to the health condition of the patients. And so it's really difficult to compare the two. And this is why we were looking for general underlying mechanism that might explain a change of power in the frequencies in the EEG. And this is, that's just an average, let's say. So the figure I show you here is just more or less an average activity, but there are always patients who show different behavior. Fine? Okay. So how can one explain the specific smile shapes? So as I already mentioned, there's an ascending reticular arousal system, and it's a distributed system over the brain, but there's an origin, and this is close to the brainstem, or there are different structures that drive the brain, that excite the brain, and the major anesthetic targets in the brain are the thalamus, the cortex, the hypothalamus, basal ganglia, and others, and also the brainstem. And so the reticular activating system, as I said, excite the brain, but is controlled. The activity is controlled by the brainstem and similar structures in the thalamus. So these, of course, are affected by the anesthetics. So our model, we thought, okay, we need the cortex because we measure EEG. So we have the cortex with excitation and inhibition. Then the thalamus plays a role, and there's a corticothalamic feedback loop. And to do this, the minimum model would be the LGN and a RTN. So this is the reticular nucleus and the relay cells in the LGN. And we have a feedback loop. So this is a known physiological structure. And we drive these areas by noise and then see what we find. And I do not provide here too many mathematical details. It's just important to understand what we do. So we have the ARAS, the activating reticular activating system that drives this network, and at the end we try to reproduce the EEG. So these are the equations that just to show you that we do not invent any activity here. So we simulated these. These are stochastic differential equation systems, and with adaptive currents. And there's external driving. There's noise from the ARAS, as I said. And at the end, we measure the EEG, and that is the randomly weighted activity in the cortex. So, and here we have the sources of the EEG is the excitatory and inhibitory activity in the cortex. So again, experimental data. So these are now two subjects of our experiments. So frontal EEG in patients under surgery. And there you see not exactly that smile, but you see it roughly. So so during the time you start on the left-hand side with no anesthetic concentration, then you increase anesthetic concentration, where you see this vertical black arrow there, the subject loses consciousness. Then you see the time scale, so 30, 33 minutes, 45 minutes, 60. After 66 minutes, the subject gained consciousness again. So it was return of consciousness again. So it took, the whole surgery took about 70 minutes. And in that time, you see there's a strong alpha. So you create an alpha here, and this alpha vanishes again. At the end, you see the strong delta on the you see a strong delta, but you see also theta activity, and but this is also, one has to be careful with this because this is also muscular activity, so from the muscles, and you measure this, and this might be artifacts. So there's a strong discussion on that. So how neural, what is it really neural, this activity, where does it come from? Because in subject 1 you see this, in subject 2 you do not see it, but you still, subject 2, you still see the strong alpha and the delta activity. So in here in the lower panels are shown the different concentrations of, I did not put labels here, but these are, so the blue, the red, and the black curves are the concentrations of 3 different anesthetics in all the cases. And there you see there are different times of onsets of high concentration levels. And so it's at the end, it's really difficult to understand this based on the concentrations of anesthetics. But I show you our simulations show this smile effect. And so what did we do to get this? At the end, all that we did is we decreased the noise level in the brain. So we started with a large noise level, so you have no rhythmic activity, and then we decreased the noise level in this whole system, and then you get the beta, you get the alpha, and then when we increase the noise level again, we reversed the whole thing. And that was really astonishing for us, because that might explain that at the end, everything might break down to the intrinsic neural noise level. You might explain this shape, this generation of beta and alpha activity just by noise, wherever this noise change comes in. And so independent of irrespective of the anesthetic concentrations and the different underlying different mechanisms, but at the end, from a more abstract point of view, maybe it's sufficient to just consider the noise level in the brain, and this immediately explains you this shape. And so of course, we took a closer look because we had a spiking network model. We took a closer look what the spiking activity did. So in the beginning, there was a very high noise level, strong noise. So you see in the relay cells, they poorly spiked, and the cortical cells were spiking, but irregular spiking. And the EEG was very flat. And when we had low noise level at later times on the right-hand side, we have a rhythmic relay activity and rhythmic cortical activity and oscillating alpha oscillation in the EEG. So in these when you take a closer look, they are bursts in the relay cells, and these bursts are well known, and they're also found in sleep. So this is a similarity between anesthesia and sleep. You find also these bursts in sleep, and the cortical activity is highly synchronized. That's also a feature. So that you find typically in anesthesia, that the EEG or the cortical activity at rest without anesthesia is desynchronized. You have a desynchronized EEG or desynchronized cortical activity, and as soon as you have a higher level of anesthetic concentration, you synchronize the cortex, and the thalamus is synchronized, and you have a very strong coherent EEG. So taking a closer look at the synchronization, here I show our results of the cortex. There you see again, so with a decreasing fluctuation variance, so from left to right, you see that there's this emerging power at 10 Hertz. Then on the lower panels on left-hand side, you see the phase synchronization. So before in the EEG, we do not have phase synchronization, but as soon as you decrease the variance, you get a strong phase synchronization. This is consistent with the findings, because general findings. So then we stumbled over a certain element, and that really puzzled us a very long time, for several months. Computing the cortical firing rate, we realized that, so that's the black line. We realized that for lowering or decreasing the fluctuation variance, the firing rates strongly increased. And this has not been found in experiments. It was just the reverse. So even the cortical firing rate decreased, but just reducing the noise made the firing rate increase, and that was not consistent with our experimental findings. And so we thought about it, and then realized, okay, we have to extend our model by synaptic effects. Of course, the synaptic effects are there. Anesthetic concentration changed the synaptic properties. And until this moment, we did not take this into account, although it's there. But we just thought, okay, can we explain everything by just reducing the noise level? No, it's not possible. So you get far, but you cannot explain all different features. So, and there are problems with this. And so we had to add the anesthetic synaptic action. And with this, we decreased the cortical firing rate, but kept all other effects. And so we realized that was a very nice situation where we realized how research really works. So at the end, you have a certain hypothesis, you apply it, you realize, okay, it might work at some day, but in some aspects, it does not work. It's not realistic. Okay, you have to extend your model, redo everything again, and then say, okay, then you can identify the feature that is essential or necessary to explain the phenomena. And so this was an additional step that we learned, that we need the synaptic action to really explain different features. And so this is what we learned. So the next steps will be other new features to explain even more features that we found experimentally. And this is how you extend your model then at the end. So, and the question was, what is the synchronization doing in the cortex? And we realized that the phase synchronization between elements in the cortex increased, as I said, but also the spike frequency coupling. So we have the intracortical spike frequency coherence. We computed this, and we've seen that under anesthesia or for low fluctuation noise and strong synaptic action, you see there's a strong increase even in the spike frequency coherence. So at the end, we reproduced the results that is known, that the intra-area synchronization increased. What we also found in the data analysis study that I showed you before, there's more information storage in each area. So, and this reflects the strong synchronization in each area. In addition, of course, based on the results in the data analysis study before, we thought, okay, the connectivity should break down, because that is the supposed mechanism at the loss of consciousness, that areas decouple when you lose consciousness. And in fact, this is the case. So we computed the spike field coherence between the cortex, the LGN and RTN, and we found that significantly this functional activity dropped dramatically under anesthesia. Just assuming that we have a high level of anesthetic concentration induces a low noise level and a strong synaptic action. Okay, and how comes that we have a denoising shift principle of the principal frequencies? So of course we had some experience already by this by our center manifold analysis before, but nevertheless we want to make sure that this is really the effect that we find here, because up to this now we have just numerical results. So we came up with a reduced model, with a kind of mean field model based on this, on our corticothalamic model, and it's coarse graining. And so at the end, I could just go through. We ended up with 4 equations, and we studied these 4 equations. And these were derived mean field equations taking into account the additive noise effect. And at the end, we have new transfer functions, Fe and Fi. They depend on the external fluctuation strength, so variance of the additive noise. And so again, these are these equations. And what we found at the end, we flatten here the transfer function, and we find a Hopf bifurcation. In fact, by decreasing the noise level, there's an induction of oscillatory Hopf bifurcation, and the frequency of this Hopf bifurcation decreases with anesthetic concentration. And we found also parameters where this is exactly the frequency range that we were modeling. And so this also describes, and these are the simulations of our reduced model. You see then decreasing the fluctuation strength, increases the amplitude, and lowers the frequency. And these are the 10 hertz at the end. You can say these are the 10 hertz rhythms that we found for low fluctuation variance. Okay, so that was we were very happy to see this, and but then the question was, how can we go on? So we thought, okay, we know there's a strong indication that additive noise might play an important role in the brain. And the question is, what do we learn out of this? So here I summarize the findings. So that first, we've seen that additive noise in multi-dimensional systems changes nonlinear actions. And how does it change it? So it changes the stability. Additive noise changes the oscillatory frequency and amplitude and the response to stimulation. And applied to neural systems, this reflects a reduced reticular activating system input, and this enhances intracortical synchronization. This enhances inter-area synchronization, and this is consistent with Tornoni's information integration theory finding, and provides insight into, in fact, provides insight into mental disorder behavior in general anesthesia. So I did not mention this before, but when you really think about it and you take a closer look at the literature of mental disorders, then you realize that there's also there are hypotheses around that synchronization and functional connectivity plays an important role, and there's so in Parkinson's disease and even multiple sclerosis, there's a breakdown of interactions between different brain areas. So there's a fragmentation in addition to local increased synchronization, and that's a very similar effect as we found in general anesthesia. And so,
Axel, I have 2 questions.
Yes.
Okay, the first one is, so you mentioned connectivity, functional connectivity between thalamus and cortex in your previous slides. So what is the measure of connectivity here?
Yes. Let me see. Yeah, here. So here the connectivity is the spike field coherence. So we were asking, so how is the field in one area? So this is intracortical. So how is the field, the cortical field activity related to the spike activity? And that's one measure of connectivity. So we use this one, that measure, spike field coherence. And And to do this for comparison reasons, we also tried to do this spike field coherence between areas, but this was a bit more complicated because then we had to compare the field activity in one area to the spiking activity in another area. So we, but we thought that's reasonable to do it, and we computed these measures, and that's the functional connectivity measure we found here. Of course, that's also an area where we should do much more work, because yeah, it's always the question, what is functional connectivity? So how do you define it? Is it just a linear correlation, as some people say, or is it more complex? Or, but you know, in my opinion, it's I'm sure you find other definitions of functional connectivity that provides you the same result. I'm also sure you find other definitions where it does not work at all. And you might say, okay, here it does not work. So, but the reason is that functional connectivity is just a concept, and to quantify this is really difficult. But at the end, for me, there's this indication that we are on the right way, because it fits together to more or less the indications or to the literature, to the finding in the last decades. You know, it fits to the picture. And at the end, this is the aim, that to describe the phenomena, explain what's going on, find an underlying mechanism, strong indication where to go, how the brain interacts. And now you try to quantify this, but we just collect evidence for a certain hypothesis, and this hypothesis is supported by our findings. And of course, we should take a closer look at the definitions of functional connectivity. For which methods does it work? For which does it not work? Why does it not work? And so on. So that's a very good question, and that's an open problem.
Okay, I have one other question. It's already 11:15, so
Yeah.
maybe one last question before we wrap up with the talk, and then we can talk for forever afterwards, so people can leave if they choose. So Scott, you have a question?
Yeah. So hey, Axel.
Hey,
so I find it really interesting that these anesthesia techniques sort of promote synchrony, but not seizure. Obviously, you know, our work together, we were looking more at, you know, these sort of synchronous oscillations in terms of how they impact seizure. This is more of a physiological, you know, alpha oscillation. So what in your research sort of differentiates the two? Is it the fact that these oscillations are sort of staying isolated in the cortex and not propagating, or is there something discernibly different between the sort of pathological versus physiological oscillatory activity that you're seeing?
So the seizure, well, let's see. Let's take epilepsy, for instance. In epilepsy, you get the seizure strong, let's say strong alpha activity or beta activity there, while the brainstem is still working. You know, it's still active, provides the brain with a certain level of excitation. And in addition to that, you have the high level of excitation, and so it's too much, and so you have these seizure waves. In anesthesia, you get the strong alpha activity by another reason, by reducing the activity in the brain, where the brainstem reduces activity, and so you get probably you get focal. So this alpha activity is typically is generated in the corticothalamic loop, and but there's no excitation from the brainstem, and so that might be the difference. That in anesthesia you see this strong alpha, but at the same time the brainstem shuts down the rest, and there's really low activity, and there's a fragmentation in the brain. While in epilepsy, I do not know whether there's a fragmentation in epilepsy, but I could imagine, I'm not sure, I'm no epilepsy guy, but I could imagine that, but maybe the difference is that in epilepsy, the different areas got synchronized to each other. And then this is a much stronger effect. And then the brain shuts down or just gets crazy, let's say, chaotic, and excites itself because there's stronger, there's still a strong connection between the different brain areas. While in general anesthesia and sleep, you lose this connection between different brain areas. So if there's somebody working on epilepsy and knows whether there's a fragmentation during epilepsy, please say so. I'm not sure, but I could imagine that that might be a difference.
Yeah, no, intuitively that makes sense. Cool.
Okay. All right, good.
Okay. So, yeah, I'm always finished. So yeah, mental disorders, okay. And very important, these are my collaboration partners. In the last, there's some more, but especially for that, these pieces of work, they helped a lot to work out these results. And I would like to point out in which city I live. Here, this is Strasbourg at a summer day, and it's really nice. So if you have the opportunity to come here, please do it, especially in the summer. Thank you for your attention.
All right, thank you so much, Axel. It's very good, super interesting talks. So we have a few questions that we answered already, but there's some more. Alexandra, first, you have your questions?
Yes, so I have a few questions. Thank you, Axel, for an amazing talk. I will start with the obvious question. Has anybody tried to wake people up from anesthesia by noising their brain, stimulating with some noisy pattern? And is that to me, that sounds like a very obvious experiment, right?
Yeah, you know, that's really, yeah, it's really interesting. So
I mean, not to humans, but potentially to animals. So
yeah, I just wanted to say, from an ethical point of view, you will have problems in hospitals on that.
For sure, but I was wondering if there was some animal research on that.
You know, currently, recently I started a project with somebody working on schizophrenia, and in a rat model. And what we do is the following. So we have a schizophrenia model in rats, and this rat has the typical features of schizophrenia. And there's a drug called clozapine that reduces these, that is also given to humans, that reduces these effects of schizophrenia and psychosis. And it seems that we get the same effect as with a clozapine as with intracranial or extracranial activation. That means, and that's a huge idea that we came up recently, is that we could, in principle, replace drugs by external stimulation. And that would be cool, because then you could avoid these drugs for patients, you know, because this is really severe for some patients. And this goes in the direction that you mentioned, that we can deal with anesthesia or induce maybe anesthesia just by external stimulation, or even replace drugs by external stimulation. So this is the future, I would say. Ask me in 20 years again how far we can.
I was just wondering, I mean, it almost seemed obvious that this could be an experimental direction. I was wondering if that's right out. I have another question about the SDF that you developed with the RTN and the LGN. The sequence in which you presented those results seemed a bit odd to me, and I was wondering how you thought about it at the time. So at the beginning, you presented the central manifold theory analysis that you did, in which you showed that you can change the bifurcation point of the dynamical system that you were studying by adding noise. You don't have to go all the way to the first slide. But then I was wondering, when you created the system of the stochastic differential equations, how did you know that they would obey these dynamics? How did you know that adding noise to that system would change the point, the bifurcation point, and the frequency of that system? How did you know that they would behave according to the central manifold theory that you showed at the very beginning? Then you need the mean field theory analysis, and it made sense because you sort of showed us that the SDF system obeys this kind of dynamics. But how did you know at the time where you created the?
You know, so this paper of Purdon, and there's another one of G. Menser and another one of Lewis, and all the and Emery Brown and all these people, and they always show a plot like this. And if you take a close look, it strikes you that there should be a Hopf bifurcation. You know, because you have a, you know, I grew up with synergetics, so with complex systems theory in physics, and their control parameter, and you know that changing a control parameter, you might induce suddenly by oscillations. And when you take a look at this picture, then you see on the left-hand side there's no activation, no frequency, and then you change a parameter, and then suddenly there's a frequency coming up, popping up, and that gets stronger and stronger. And Jeremy and we looked at it and we said, you know what, there should be a Hopf bifurcation, but why? You know, there should be one, and we knew that we could, well, what I did not show, but we knew that we could induce Hopf bifurcations by additive noise. And so the idea was that, well, what is it if we can describe this? You know, just explain. Then we did not know why this should make sense. You know, we just came from a mathematical side.
Were you confined at all by the physiological parameters of your stochastic differential equations, the ones that you had to choose? Was that like a limiting factor?
You know, that work I show you now in an hour, that's a work of 5 years, really. So I know at least for 3 years, we were looking for a parameter set that makes this thing work, you know. And at the end, we were happy to find it. But we thought it should be there, you know, because we know if you have a nonlinear system and you couple it with noise, then we know we change the bifurcation point. It's there. We understand that it's there. It's clear that it's there. You know, you change the synchronization. We did this study for many different simpler models, and then we know it's there. But where is it in this huge parameter range? And then we played around, and Jeremy found that at the end, he came up with a Debtgen currents, and that really also helped to get this effect, you know, without the adaptive currents. Let's see, where do I have it? These adaptive currents then here. This does not work, and these adaptation currents are necessary. They're there in physiology, you know, but we needed to get this. And at the end, it worked out, you know, and at the end, you say, okay, these are the features. Now we know which features we need to get this effect, and they're all physiological.
Interesting. Okay, 2 more questions, hopefully quick. It was interesting to me to see that the EEG is becomes flat at page 22 as you get into anesthesia. That just made me realize, irrespective of anesthesia, that this shows that EEG reflects cognitive functions only, or almost for the most part. I mean, obviously the organs are functioning and the heart activity is still on, and all this vital activities are still reflected by some kind of brain activity. They map to the brain, but they're not reflected in the EEG. So does that tell us that the EEG essentially only reflects cognitive functions and not the activities of vital organs or anything like that? Anyway, that's maybe a side.
You know, in this case, a very deep anesthesia, when you have a flat line, so this is coma, you know.
Mm-hmm. Right.
And
so maybe that delta activity that you had shown is what?
Yeah, so this, if you have a flat line, then this is almost, well, this is not pathological coma, this is reversible coma. So if you really increase the anesthetic concentration more and more, you know, this happens also for remifentanil people in US. Now recently they came up, it's a huge wave of people taking remifentanil, and they killed themselves, you know, because it was too much. And so if you take too much, then yeah, you get heart arrest, and that's it. And if the anesthesia is too deep, then your brain stops working. You know, there's no interaction of any areas anymore. And then this is well controlled here. This is an experiment under the lab conditions. So it's reversible. But you can also, there are several studies of coma patients who had an accident or something like this. And you see also a flat line. And so this is the case where the patient cannot survive on his own anymore. So,
Alexandra, I have a few people in line for a question.
Sure, I'll stop, yeah.
No, but I mean, we can come back to you just to...
Sure.
Mauricio, you had a question.
Yeah, can you hear me?
Yes.
Okay, hi Axel.
Hi.
Maurizio here. So I really like this idea that our consciousness is just a nuisance, so nice. I have nevertheless just a remark. So if you go to the slide of the figure of Bourdon, right, on PNAS, so in the smile effect, yes, so I could not avoid to notice also that there is this increase in power on the slow rhythm. So how do you explain these additional oscillations in the ultra slow rhythm? Are they like an artifact, or are you going to take into account this?
Yeah, so our model does not take into account this. So we were focusing on the alpha activity, and there's a strong debate where this alpha, this slower 1 Hertz rhythm, it's also called smaller 1 Hertz rhythm, delta activity, where this comes from. And currently it seems that there's a consensus that it's cortical. So it's not corticothalamic, but it's cortical. And it's not clear yet where this comes from. It's there. It's also there in sleep. That's an interesting point to know. And sleep is also, and it emerges also only when you sleep, and sleep is driven by the brainstem. And that's another indication that maybe the activating arousal system plays a major role in the generation of this delta activity. But we were not focusing on that one, because we were aware that this is missing in our model. But that would be also the next step to, but I'm sure that we need at least another area, not only the corticothalamic feedback loop, but maybe another, another model or additional extension of the model, taking into account the cortex, intracortical activity. And maybe that might explain the delta activity. So currently I'm working on that, and I have first ideas about that. But of course, that's a missing point here. But and they are strong. I can also mention that there's also debate on the coupling, on the frequency coupling, or the frequency phase coupling between delta and alpha activity. And some people find it, some people don't find it. And it's still not clear where this comes from. What is the role of the delta activity? But definitely it's always there when you lose consciousness. This is for sure.
Okay, thanks.
I have a question from Henri.
Okay, thanks for the really interesting talk, Axel. What experiments do you think would need to be done to verify that the output of the reticular activating system behaves in the way that you need it to behave to vary its noise? Is anything known about that, or that could be done about that?
Experiments. Yeah. Well, it's known
like
that
in animals, or anything. Like, is there a way to measure this noise and to see, yeah, the ARS goes up, there's really this change in noise?
Well, there are very few. I checked the literature in detail, and there are very few studies showing the properties of this reticular activating system activity. And there's one study, only one I found, that they say that there are electric synapses playing a larger role, and they found some way higher distributed higher frequencies in the gamma range. That is a property of this activating excitation signal to the brain. And there's a hypothesis around that electric synapses play a large role. But this is all that I found. And the problem is that the reticular activating system distributes widely in the brain, and there's not a single center, but it activates the whole brain, and so it's hard to really identify this. So,
and for you, it went into LGN and RTN, but did it go? I can't remember. Did it go to cortex as well?
Yeah, definitely.
So it was activating all three.
Yeah, all three, basal ganglia as well, in all areas. All areas, and there are even feedback loops. So from this area to the brainstem and so on. But what is known is that it's really there, and that it really drives the brain. And the origin is in some way the brainstem and certain areas of the brainstem. And it's known that these areas change the activity characteristically when you lose consciousness. So then we are back at the switch, at the sleep switch. And so this is known, definitely, and this is for sure. Whether this is noise, I just say noise, this is Gaussian noise, independent, equally distributed noise. Okay, this is, I say noise because I do not know the features, and so I call it noise. Of course, it's possible that this driving activity has a different spectral distribution, and maybe gamma frequency is very important to drive the different areas. But in the moment, since nobody knows what's going on, and it's just known that it drives the brain, we say it's noise. It's white noise.
I have another question from Francis Skinner. Francis. So can one implement interacting drug effects in the model to test the modeling aspects?
Yeah, we did this by introducing this synaptic action, this anesthetic synaptic action, where you just on the GABA A receptors. And this is, as I said, this is mandatory to explain all features. We did this for propofol. So there are other studies doing this for isoflurane and halothane and other drugs. But this is really, of course, you can do this, but it's not known. The on-site concentration at the synapses is not known, and it's not known even the distribution of GABA A synapses is wide, and they are distributed all over the brain, and so they are targets of the anesthetics. There's even study of subtypes of the GABA A receptor, and even there you have wide distributions. So it's really difficult to say what is the on-site concentration at the different synapses, since the system is too complex. And this is why we, at the end, when you think about it, we said, okay, let's maybe go away from this idea to model single drug effects. Of course, that would be nice, but since we do not know how the effect is distributed over the brain and how strong this effect is, it's really tough to get anything working or to get any insight. And so as usual, as in science or in physics or in neuroscience, we go to a higher abstract level, higher description level, and say, okay, maybe we understand more the mechanism, the major mechanism, and maybe that already helps to understand what's going on. Well, we still do not know what's going on the microscopic level.
Yeah. So, sorry, can I just expand on that slightly? Like, so I appreciate that, and I mean, this is where it's really cool. Sorry, oh, there's sort of noise. But I guess it's sort of like trying to sort of take advantage, right? So it's sort of building on what Andre was asking about, sort of testing or linking with experiments in some way, right? So you have the advantage of sort of this abstract high-level model, right? So it's not so much exact concentrations or distributions with GABA A and these different things that are known with axial latex effects and all these, you know, detailed molecular kind of studies, but more just a sort of general principle. If you can kind of test it, you know, by different levels of noise and interactions that would come about. So sort of the general principle that you're proposing about the anesthetic effect is tested. So that's kind of what I was getting at, but it might be an ignorant question on my part, and I will mute so that you can hear. Yeah, that's what I was asking. So there's not really an answer, but that's, I thought to take advantage, if you like, of your abstract high-level model. Yeah.
You know, at the end, it's a very good question. So at the end, you want to understand, well, it's nice to understand how the brain works, but at the end, you want to go to the clinical application. So that's the major aim of the research. So what is it good for? So what do we learn about the brain now? How does this help anesthesiologists? You know, how can we improve general anesthesia now with these findings? These are all the questions that at the end we try to attack, and that's a good question. So. And I think the next step would be definitely to understand better what each anesthetic drug does. How does it contribute? You know, of course, that's a good question, because then this also gives you advice how to develop new drugs, maybe for better effect, that have better, less side effects. Of course, that's an important question. But even already understanding that at the end, it might be the brainstem, you know, that triggers all this. Of course, there are synaptic effects, but at the end is the brainstem, the reticular activating system, that triggers all that. Maybe that provides a focus, experimental focus. Maybe I already thought about how is it possible maybe to just change the activity of the brainstem, just that one. And if we find an experimental technique how to change this one, then we could make a test of this hypothesis, because I'm sure when we can control just the brainstem, then we can test whether this is true, whether we could induce unconsciousness just by changing the activity of the brainstem. I think that's the central part.
And also, I mean, I'm just going to add, like, if I may, Axel, I think also that for this work, there was most of the modeling done before anesthesia was clearly and only cellular based. It was a very neuron synaptic effects only were modeled, and there was no circuit based effects, right? And this is kind of a shift of scale too, just to say that there are probably different contributors to the effects.
Yeah.
Okay, so I think we kind of busted time a lot today, but that was a very interesting talk. Thank you, Axel, again. Yeah, so for those who remain here, we have a KCN event on December 10th. So it's going to be Andrea. Hold on, I just don't want to mess the name up. Diaconescu, who's going to be presenting her Bayesian hierarchical models. So that's going to be on December 10th. Details to follow by email. Okay, so thank you so much, guys, and see you in a few weeks.
Thank you.
Okay, shall we chat, Axel?
Yeah.
Okay. All right. Goodbye.
Hi, everybody.
So I'll send you a Zoom link, Axel, by email.
Yeah.
Okay.
Oh.
Bye, Axel. Thank you very much.
Right.